Perfect. Thank you for having me here to talk about the Canadian BioGenome Project in the Earth BioGenome Initiative. We have been working with a large group of outstanding scientists and scientific leaders, many of which are in the room today, that have come together to help put this project in motion as the Canadian branch of the Earth BioGenome Project. That's working. So the Canadian BioGenome Project, or the CBP, is part of a global initiative called the Earth BioGenome Project. We are one of about 54 affiliated project networks across the world, and the goal is to create annotated chromosome scale reference assemblies for at least one representative species of each taxonomic family. And we at the CBP are here to make sure that Canadian... Should be. Will that be better? Is that okay? Better? No? Okay. Oh, really? Is that better? No? Just hold it? Okay. And I do have a question about the slides, just scrolling to see my notes. I can't seem to do that. Not overly important, but just that part. Okay. I'm not that through it. Yeah. All right, that's fine. Anyways, so the goal is to create an annotated chromosome scale reference assembly for one representative species for each taxonomic family. So we at the CBP, we take care of the extractions, the sequencing, the assembly, and the annotation. We have an open data sharing policy, so we will upload the data to NCBI. We also provide free shipping for any of the samples that are collected, collection tubes and reagents, anything that you guys need. We have 2 extraction facilities and 3 sequencing nodes in Vancouver, Toronto, and Montreal. So we have established about 32 taxonomic committees or panels of experts, which are helping us to select which species in Canada we'll be sequencing. And on Friday afternoon, myself, Pam, and a few other individuals helped put together a workshop to select which reptiles and amphibians we will be sequencing for this project, as well as identifying potential users of the data and sample collectors. So if you do have any additional suggestions or feedback, then please come find me at the end of session 8, or you can send me an email, which is on the bottom of the screen, and it'll be again at the end of my slides. So how do we determine which species we'll be sequencing? So the Earth BioGenome Project's provided a few recommendations. Is it publicly registered? It has a tax ID. There's no known species complex. Some other considerations that we take into account are if it's a threatened or endangered species, if it's an invasive species, if it's found in the Arctic or subarctic, as well as the likelihood of obtaining a sample. So we have been working very closely with Earth BioGenome Project and the Darwin Tree of Life and the experts on the various committees and panels to identify suitable sample collection protocols for each of the 32 groups. Some things that we take into consideration are the number of specimens and the size of specimens that we'll need for collection. So will we get enough material from one fish? How many spiders do we need for sequencing of the 3 technology or platforms that we'll be using? The location and duration of field collection are also very important. So will you be collecting specimens for 1 hour versus a 3-week expedition in the Arctic? That ties directly into your access or availability of equipment to prepare, dissect, and preserve the specimens. So if you're collecting bees and you're about, 5 minutes from your lab, you can probably bring that specimen back to your wet lab do the dissections. You probably have a -80 or -20 freezer, and you can store the samples in there in a relatively short turnaround time. Whereas if you're in the desert, for example, you probably don't have easy access to a -80 freezer or dry ice or liquid nitrogen or anything like that. So you have to get a little bit more creative with how you are preparing and preserving the specimens. Shipping. Standard, we use FedEx overnight priority shipping. And again, we do cover the costs of shipping samples to us. So very briefly, I'll go over the sample collection guide for reptiles. As I mentioned, reptiles and amphibians. I mentioned earlier, we have unique collection guides for all of the 32 taxonomic groups. So we try to list all of the possible preservation and storage methods based on what equipment you guys might have. So the gold standard is always blood whenever possible, preserving an aliquot in an EDTA collection tube, another one in RNA protection tube. And for high C, we recommend putting about 1 to 2 drops of blood in about 1 ml of 95% ethanol. For smaller species where you may not be able to get adequate blood, then we do accept dissected tissues. These can be preserved in RNA later or ethanol, and we always recommend that they are flash frozen and stored in a -80 freezer. Specifically for RNA, we encourage getting a diversity of tissues from different organisms at different life stages, from different geographic areas or designatable units. So once we receive the samples, we also collect robust metadata, high resolution images, voucher specimens, and we also get the sample collector to acknowledge the data deposition policy. So in parallel with the EBP and the Darwin Tree of Life, we have an open access data sharing policy. So all of this data and information will be freely accessible to anyone who wants to use it. And because we are part of a larger sequencing initiative, all of our data and information is tracked on various websites. So you can see which samples have been collected, where they were collected, specifically which country, what the status is, have they been extracted or sequenced or assembled. Excuse me. You can also look at QC reports, various assembly metrics through one of the various data sharing portals. So we plan on sequencing about 400 to 460 genomes. As of about a month ago in mid-August, we have collected just under 1,200 samples for 97 different species, and we have performed over 300 extractions for RNA, DNA, and our high-C chromatin capture. And I've put a picture of our progress bar, which you can find on our website. So we are using 3 main sequencing technologies, our long-read pack biosequencing, our high-C chromatin capture, and RNA-seq. Specifically for RNA-seq, we're using that information to annotate the genome, and we are doing, in some instances, a combination of both long and short-read transcriptomes. So as of mid-August, we had generated over 7 terabases of data, with half of that being in the last 6 months. And most recently, in the last 3 weeks, we've generated an additional 2 to 3 terabases of data, which is not displayed here in this figure, but I will but I will show some of it in a few slides. So we have been very busy collecting samples and progressively ramping up our production and data generation. So we track each species on our project website, as well through various EBP websites, such as Genomes on a Tree and the Darwin Tree of Life. So this is a snip of part of our project status on our website. So it's got the progress bar, so you can see how many samples are being extracted, where they are in the sequencing and assembly queue. We also have a list of the species at the bottom with direct links to NCBI, where you can view and download the data. So to date, we've created bioprojects for 46 species, 128 biosamples. We've submitted 105 data sets to SRA and 24 genome assemblies. The 24 assemblies include 14 primary assemblies and 10 alternative or alt haplotypes. This number doesn't include mitochondrial assemblies, which we also take care of. So this was our poster child for the CBP. It was the first species that we sequenced. The data was released on NCBI a little over a year ago. And I think 8 months after the data came out, we submitted our first genome note, which is available online. And this genome note basically just details the sequencing and assembly methods, and then gives a cool comparison of the loggerhead sea turtle to the green sea turtle. And it only gets better. So this data was fresh off the Revio sequencer about 3 weeks ago, and we've put together 5 genome assemblies in a relatively short time. 4 of them are displayed here, and it looks like about half of them have already been uploaded to NCBI, and the other half are pending submission. And the typical turnaround time until the data sets are viewed on NCBI is it's a 4-month backlog. So if anyone would like access to this data before it's available online, then please just send me an email or reach out through our study website. And we also submit the mitochondrial assembly and annotation as well. So I won't go very much into detail on the annotation pipeline, but it is spearheaded by the European Bioinformatics Institute, and they are taking care of the annotations for all of the 54 affiliated projects across the globe. So the Wellcome Sanger Institute, they have an internal platform that they've been using to publishing genome notes. As of December 2022, they had about 5 genome assemblies that were completed. And most recently, they just passed the milestone of 1,000 genome assemblies. So they've been working with a few other project networks to automate the production of a genome note publication. So to clarify, a genome note is a brief description of how and why the genomes were sequenced, and they don't include any analyses or conclusions. And a bunch of really cool figures and tables are generated from this. We always like to throw in a Jupyter plot at the end, which basically just compares the new assembly to a close relative. So that should be live in the next 6 to 12 months. So progress to date for reptiles and amphibians. We've had about 16 species in various stages of extraction, sequencing, assembly, and annotation. It looks like just the loggerhead sea turtles made it all the way through to completion in terms of annotation. We have about 5 assemblies that have been ready in the last, I think, week or two, which will be uploaded to NCBI shortly, and about 10 other species that are currently in various stages of sequencing and extraction. And this project wouldn't be possible without a lot of outstanding scientists and scientific leaders, not just within Canada, but around the world. Thank you.
Okay, awesome. Wonderful to be here today to talk about this truly collaborative community science project. I'd like to start by offering my gratitude as a guest here today at Carleton University on the traditional and unceded territory of the Algonquin Nation. This project includes community science observations from the traditional territories of diverse First Nations and Indigenous peoples across what is now called Ontario. And historical and ongoing Indigenous stewardship of these lands has been essential to the conservation of biodiversity across Turtle Island. I want to acknowledge that relationships with and observations of reptiles and amphibians existed long before written records. And so with that in mind, this project that I will be highlighting offers a culturally specific perspective about our understanding of reptiles and amphibians in the province. So Ontario Nature is a charitable conservation organization that was formed in 1931. We protect wild species and wild spaces through a variety of programs which combine conservation, education, and engagement. One such program is the Ontario Reptile and Amphibian Atlas, which has engaged the general public in collecting reptile and amphibian observations since 2009. Atlases are projects that collect observations of a target species group from many sources in order to improve our understanding and knowledge of the distribution of those species. You may be familiar with the Breeding Bird Atlas, for example, but there are many examples around the world covering diverse taxa. These atlases are a valuable source of detailed species occurrence data that can inform research and conservation work, and the resulting publications provide a wealth of information about the species, including detailed range maps and information on the identification, ecology, and behavior of the species. Often this gets saved for the end of presentations, but I really want to start by acknowledging the many, many contributors that made this project possible. The Atlas is a huge effort that includes observations from more than 12,000 contributors, including a great many people that are here this weekend. And I'd like to just give a special shout out to the regional coordinators, the organizational champions, the Atlas Steering Committee, and the Publication Steering Committee, who were essential to not only support the data collection phase of this project, but also the many people who were authors, reviewers, and more during the preparation of the publication, which is still ongoing. So I stand here today as one person, but I'm really representing a large collective of people that have contributed. The Ontario Reptile and Amphibian Atlas was not the start of the Atlas efforts of herps in Ontario. The original herp atlas was the Ontario Herpetofaunal Summary, or OHS, which ran from 1984 to 1995. As the first species atlas in Ontario, the Breeding Bird Atlas was winding down. Naturalists were looking for another project, and a group of enthusiastic individuals decided to start a project on the distribution of reptiles and amphibians in Ontario, and thus the OHS was born. 3 reports were published over this atlas period, and over 140,000 observations were collected by more than 4,000 participants, leading to the first comprehensive understanding of reptiles and amphibians in the province. Reptile and amphibian distributions in the province. However, by the mid-2000s, the OHS data was becoming outdated, and there were still relatively limited in their geographic scope. So while several hundred records continued to be submitted annually by a handful of contributors, there was little concerted effort to engage the public in ongoing data collection. And so in an effort to ramp up this engagement, the Eastern Ontario Herpetofaunic Atlas, or EOHA, was launched by the Eastern Ontario Model Forest in 2008, similar to OHS, but focused in Eastern Ontario. The EOHA ran for 2 years and had really important accomplishments, including developing the program framework and methods, outreach materials, website, data reporting forms, which really all helped to recruit new contributors and rejuvenate data collection efforts. Another key focus of the program was to solicit herp occurrence data that had already been collected by researchers and other organizations. With the initial success of the EOHA, work began to expand the atlas, and Ontario Nature was approached to support this effort, ultimately expanding the atlas under the ORA name. As was similar to earlier atlas efforts, outreach and engagement remained a core focus of the program, with the ORA soliciting observations from community members, researchers, and organizations province-wide. Data collection continued until 2019, and since then, the focus has been on preparing a publication of the results. And I'm very excited to stand here today and tell you that that publication is very near completion, with an anticipated release date later this fall. The overarching goals for our upcoming publication is to highlight trends that have been found over 30+ years of data collection across all 3 atlas efforts, and to use it as a tool to continue engaging the public in herp diversity, threats, and conservation issues. We also want to use this immense data set to inform conservation efforts moving forward, as understanding where reptiles and amphibians exist is obviously crucial to their conservation, allowing for the identification of important habitats, threats, and potential hotspots, and to assist with assessing species at risk and developing recovery strategies. So how did it work? Similar to the Breeding Bird Atlas, the province was divided into 10 by 10 square kilometer grid, and observations of reptiles and amphibians were then collected from a wide range of sources, with each observation being associated with a specific grid square. The effort varied across the province and across records, which ranged from intentional standardized surveys to one-off opportunistic observations. Observations were collected from all different types of folks and groups, and they were accepted through all different types of means, from people literally phoning us to call them in, sending us mail that had lists of their species, and then later on through the program, much more common emails, and eventually there was an app developed which really helped with online submissions. And people would submit everything from a single observation to massive spreadsheets of many, many observations of herps. And then all those observations were verified for identification and location accuracy by Ontario Nature staff and by volunteer experts. While our approach to data collection had far-reaching impacts on the scale of data collected in the province, you can see there's still a varied effort across the province. These maps here show the number of observations in the Herp Atlas by square, with the lightest color representing squares that have less than 100 observations, and that darkest color representing squares that have over 15,000 observations recorded. Over the lifetime of the atlas, we have a cumulative database of 480,000 observations, including about 250,000, which were collected through the 10 years of the ORAA. The success of the atlas has largely been from the immense efforts of, again, the over 12,000 people that contributed and over 200 organizations across the province that partnered on this project. And of course, the growth of the Atlas since 2009 would not have been possible without the efforts in engaging the public, which through over 260 reptile and amphibian focused events, Ontario Nature engaged over 27,000 people. This is a rough breakdown of the species observed by group in the Herp Atlas. And so I want you to think it in your head or shout out what you might think is the species group that fills the largest piece of the pie here. Any guesses? Frogs, yes, frogs and toads. There's about 293,000 species of frogs and toads contributed to the atlas. Any guess on the next largest slice? Snakes and lizards make up the next largest at 86,000 observations, followed very closely by turtles at 79,000 observations. And finally, perhaps unsurprisingly, that small piece of the pie there, salamanders at 24,000 observations. They're lumped in with snakes, yeah. Herp Atlas efforts since 1984 have certainly helped our understanding, helped to improve our understanding of the current species distribution in Ontario. So this is particularly true for some species at risk, including the Blandings turtle here, where you can see this is our knowledge as of 1998 with about 1,700 records of Blandings turtle, to here in 2018 with over 8,000 records being added over the last 10 to 20 years, and over 300 grid squares that have had recent observations of Blandings. So that means within the last 20 years. We've also greatly improved our knowledge of common species as well, as seen here with the eastern redback salamander. In 1998, we had about 3,600 records of these guys in the atlas. And here we are in 2018 with the number of records doubling, and over 300 grid squares now have recent observations observed within the last 20 years. So now, of course, the important question is, what can we do with this immense data set, and how can we use it to inform conservation efforts moving forward? So here's a list of different examples of data requests that have tapped into this database. They're used for species inventories, threat mitigation, academic research, all sorts of things. And I think it's also important to highlight the contribution this project has made in increasing awareness and enthusiasm about herps in the province. So if you're interested in using this data to inform your conservation work, or if you're looking for a collaborator for projects that address threats or knowledge gaps for herps in Ontario, definitely reach out. I'd love to talk to you about how we might be able to work together. So outside of showing you how the Atlas data can be used to direct conservation action, I hope I've also shown you the value and the power of community engagement in conservation. While Atlas efforts since 1984 have greatly improved our knowledge of herps in Ontario, continued community engagement and stewardship will be essential to herp conservation in the long term, and continued submissions of observations, particularly so in areas where our knowledge is still lacking, is very important. There is still a lot to learn. So while Ontario Nature is no longer accepting direct submissions of observations, we do strongly encourage people to continue to submit your sightings of herps in Ontario to the Herps of Ontario iNaturalist project or NHIC's Rare Species of Ontario project, which can both easily be found on iNat. Okay, so what can you expect from the publication? It will be freely available in online format, and there'll be an associated story map as well online. And it will include a wide range of topics, including a much deeper dive into the introduction, the history, and the methods than I went into here today. And also detailed species accounts for all native herps in Ontario, and bonus coverage on non-native, extirpated, and unconfirmed species in the province. There are conservation chapters, including on threats, the impact of this atlas, and multiple research articles and short articles, such as one I highlighted here on the effect of habitat loss and roads on reptile occupancy, which was written by James Patterson and Christina Davey. So just one example of a lot of the great articles that you can find in the Herp Atlas. There's also sections on future directions, where do we need to go, and a whole section on stewardship, how you can help herps in Ontario. There's also about 50 pages of acknowledgements in this publication, which really just recognizes the collaborative nature and how many people contributed to the project over the years. So I'm excited to give you a little bit of a sneak peek into what the publication will look like. It's currently in design phase. I did grab some screen grabs for you here today. So you can expect the ORA publication to be full of beautiful photos and highlight the conservation status, phenology histograms by species, and the most up-to-date range maps for herps in Ontario available. There are details on the appearance, biology, habitat, and trends for each species. And there's a whole whack of interesting topical articles on herps. And again, 50 pages of acknowledgements where you can look for your name in there if you contributed. So some final notes then, save the date. We'll be having an online webinar and virtual celebration to launch the publication on November 29th. We hope many of you will be able to join us. And next spring, we also plan multiple in-person events to be able to go out, look for herps together, and really celebrate the success of this project. If you feel so inclined, help us cross the finish line. We have some design and editing costs that we're trying to cover. So go to ontarionature.org and make a donation to help us finish this project. And if you'd prefer a hard copy to the free online version, we will be doing a limited pre-sale of hard copies. So if you're interested in that, keep an eye out or get in touch with me. We don't know exactly how many we're printing yet, but the more interest I know there is, the more likely we'll be able to print more copies. So definitely reach out and let me know. And if you know of libraries or institutions that might want this book added, definitely share the word and let people know that it's coming. And so I'll leave it there and just thank the many funders and again, the many people that contributed to this project. And I'm happy to take any questions.
So this is going to be a very, very broad overview of an awful lot of work. So some people won't be familiar with this project if you haven't seen previous presentations over the years. So I'll start with a little bit of background going back to the early days. This was an effort to look at turtles in Muskoka, particularly Blanding's turtles. In that year, the ESA regulations kicked in to have habitat protection generated by observations of Blanding's turtles. And so we want to trigger a whole bunch of that. And we also want to study nest predation with respect to anthropogenic factors that might influence it. And so that was a partnership with Laurentian. We got started with Jackie in that year. And Hannah McCurdy Adams was a grad student who helped us launch this work. Okay, so for those of you unfamiliar with this, if you put a dot on a map for Blanding's turtle in Ontario, then you trigger habitat protection in a 2-kilometer radius of suitable wetland habitat and also a terrestrial buffer around it. But if there's no dot on the map, there's no protection. So we want to trigger that in Southern Ontario. This is the area we started to work in back in 2013. And this wasn't randomly chosen. It's partly chosen because it's close to our center, the little star there at the north end of Lake Simcoe. But also this area is kind of representative of the southern edge of the Canadian Shield all the way across Southern Ontario, what's sometimes referred to as the land between. And we thought that it would be useful to study that area particularly because all the other long-term studies of turtles were up in Algonquin Park or on the Georgian Bay shoreline. And those areas are very different in terms of threats and climate compared to the rest of Central Ontario. So if we base management decisions on those areas, we might be getting it wrong. So remember, this is all back in the very early days. After the first year, Canadian Wildlife Federation and the Rogers Foundation came on as really important partners and provide us with quite a bit of money over the last 9 years. And the project got a lot bigger with lots of partners and a lot of effort going into it. In terms of expansion, just before the pandemic hit, that blue outline is where we were currently permitted to work. How did we do for Blanding's turtles? Well, we found quite a lot of them. And we triggered a lot of habitat protection. We're over 5,000 square kilometers as of 2022. We haven't crunched this year's data yet. We have done a lot less work this year. Our funding's been very much interrupted this year. Lots of challenges there. So we haven't added much to that probably for this year. One thing that is really interesting to me with a large scale, like landscape scale project over a long period of time is we can pick up some things that you wouldn't find working at one study site or working in a bigger area for only a couple of years. We talked a lot about how turtles show site fidelity, come back to the same places all the time, and we do see that a lot, but sometimes they don't. Sometimes they pack up and move, just like people do. And so this is a female turtle that was captured nesting 30 kilometers away 4 years later from where it had nested before. And so you can't find this kind of stuff with short-term and small-scale studies. And we've seen spotted turtle movements of 6.5 kilometers as well. Speaking of spotted turtles, we found a lot of those. We've identified 20 new locations for spotted turtles in Muskoka. After somebody told me we wouldn't find any there, I thought that was kind of funny. One thing you might take from this graph is that spotted turtles are a lot easier to capture than Blanding's turtles. We don't miss very many of them, except in 2022, you can see it went up there. That's because of a radio tracking study that we launched. And so we're not catching all of those turtles all the time. But you already heard from Steph about his work with the coloration trials and stuff. But part of it is also a radio tracking study. So those early years where we were looking at nest predation, we found it to be about 80% in Muskoka. And so as a result, we increased the nest caging work that we were doing. And we did also launch a nest excavation and captive incubation program because we can't cage nests everywhere. Some of them are in the middle of a dirt road. And what came out of that is that if you place a lot of nests on the shoulders of roads, the people who make the shoulders of the roads and manage them don't like it very much. And they told us to stop. And so we ramped up our captive incubation program, which led to this. And so last year, we incubated our 100,000th egg. And we didn't do too many more this year. It's really Kelsey and Sarah you should be applauding, not me. But we're over 118,000 eggs as of this year. Oh, almost 120. We haven't put this year's data into it a lot, so I just like tucked little numbers on for where we're at currently. But that's our egg incubation numbers over the years. We started head starting because we got some Blanding's nests south of the Canadian Shield. And as you saw, we find a lot of Blandings in the Canadian Shield, but the number we find that are south of that, like down towards the Barrie area, is very, very few. So we started head starting. We're head starting about 100 Blanding's turtles a year now for Simcoe County, but also for Royal Botanical Gardens down in Hamilton And this may ramp up with some potential funding that might happen. We'll see what happens with that. It takes a lot of people to make all this work. So our team's actually gotten bigger since then, but apparently we suck at taking group photos now. And so it was almost twice that big through the pandemic. And of course, you have to train all these people. So we launched Turtle Boot Camp, which is now Reptile Boot Camp. And it's a very intensive 2-week training program all about field methods related to studying turtles in Ontario. And then we set all these people free to go out and do all the field work. But we also engage the community a lot, because no matter how big that team was, it's still spread very thinly across such a huge area. And there's always more of the public than there is of us. And so we approached the government to get approval to train community members who we would then add to our permits. And so our permit list is like 120 people long at this point, most of the years. But we train these people to do varying levels of things, from simply transporting a turtle legally because they couldn't otherwise, to actually being the person that we call and say, hey, we want you to go over here and dig up this nest because it's just down the street from your house, and then we don't have to drive there. And they do that. And to be honest, I was surprised when they said that we could go ahead with that, but we got the buy-in, and it was great. We launched this program a few years ago during the pandemic to be even more advanced training. It's basically all the stuff that I thought that students are not getting out of college and university programs to help them with their careers. And that worked really well for a couple of years, but it's been on hiatus now, partly because we have to rebuild our instructor team after we hopefully get our funding situation sorted out. On the community engagement side, we launched the Turtle Hotline, which was a very useful tool for us. People phone us and tell us about their sightings, hopefully of what we call rare, nesting, injured, or dead turtles, not necessarily every painted turtle they see at their cottage. Because all those things are really useful data points for us, but this isn't just a reporting tool, because when our dispatcher takes the call, they decide whether to send out a team. So if you call us with a Blandings turtle in your front yard, we're sending someone to your house, most likely, if you want us to. But a lot of people do. And it's a really useful tool because it actually gets us into people's lives and engages them with it. They get to name the turtle, and they also invite us to survey the pond out behind their house. Or some of them own like 500 acres and half of it's wetland, and we couldn't get on their land before, but now we can. It's really cool. Here's our calls graph. So we're probably going to break 1,000 calls this year. We do a lot of driving for this project. It's a lot of nesting surveys along roads. So we've got hot spots identified where it's a bit of a problem, obviously, for mortality. We want to dial in on that a little more. So a few years ago, we launched these 1 kilometer walking surveys that are repeated every week to try and refine the hot spots further. And so we get a lot of data through that. We've stopped doing them after the last few years, though. But it's not just about mortality monitoring. We want to actually do something about this. And so, of course, we could do some fencing. But in Muskoka, it's a very continuous habitat. You would have to fence dozens of kilometers of almost every road to keep turtles off of the roads effectively. So that's probably not going to happen anytime soon. But what we can do is put people on the roads, not literally, hopefully in most cases. So if we see that snapping turtle that's on the shoulder nesting, and we put somebody there for the next 3 to 6 or more hours that it takes until that turtle's done and walks away, we make sure it doesn't go on the road and get hit. We've effectively stopped that turtle from becoming another statistic. And if you do that often enough, hopefully you have an effect on road mortality on that particular road. And we'll, of course, also find turtles just walking across the roads, and we move them off the road, that sort of thing. So if you can imagine, if you scaled this up enough, you could have enough people on this road that no turtle would ever get hit by a car, which would be difficult. But in theory, it's possible. And so here's Housies Rapids Road in Muskoka. And as our search effort scales up on the road, you should see that the number of dead turtles starts to level off, or maybe even starts to decrease. And in terms of proportions, the proportion of live turtles should go up and the proportion of dead turtles should go down. And that's exactly what this road shows. Because some of these roads were on them 5 or 6 times a day to get up to these higher levels of search effort. Of course, not every road is as nice as this. And if we looked at Highway 11, you'd see that it has no effect at all like that. And effectively, there's so much traffic on Highway 11 that even with 1,000 people on the road every day, we wouldn't get to a turtle in time to stop it from getting hit by a car. So this kind of mitigation wouldn't work on Highway 11, needing fencing. But on Hosie's Rapids Road, maybe this is a good approach and possibly cheaper than fencing and maintenance. We launched map turtle work for about 3 years because our funder asked us to, and it was great. And that's a story for another day. But by last year, our area had grown more. We added new partnerships. We were doing a lot more excavation of problematic nests in different places, and they just kept amending our permit all over the place. And we're going to move on to snakes. So 3 years ago, we launched the CARES project, and it's focused mostly on Massasauga rattlesnakes so far, but the other 2 species are of interest for us as well. And although we had a bunch of goals, the 2 big ones for me that I really was interested in was to try and better delineate the eastern and southern boundaries of this species as they are today, not what they were historically, and also to go out and identify confirmed hibernation and gestation sites, because those enable long-term monitoring in a better way than road surveys and transects and things like that. And so here's what we found. We've got a whole bunch of confirmed gestation sites, hibernation sites. We've caught a whole lot of snakes, which has, of course, enabled some other things along the way. So how do we do with those boundaries? We're still working on it. That red line is a very, very crude, like I literally drew it on my slide last night, estimate of what seems to be the eastern and southern boundary currently. But you can see there's some historic areas that seem to be lost. We have not, in even the 10 years of start, but especially these 3 years of intensive stuff for rattlesnakes, we've not got any observations from us or from the public out in those areas. And of course, we've been able to support some research. This is not a research project overall, but there's research that goes on within it. And you've seen Meg's talk, hopefully, or Ginger's already today. But this is stuff that's being supported through the CARES project, the SSP plan for Massasaugas with captive breeding efforts. That's our facility for that. There has been some publications that came out of the work so far, and there's more to come, especially hopefully with those 3 grad students. But Kelsey has one in prep, I think, now, and so there'll certainly be others. And there's a lot of people, so I can't list all their names because there'd be like 300 people on there at least for all the various people involved. But I did put some especially helpful people on there to say thank you to them and to our partners and funders and everybody else. And hopefully this work will keep going. And we are very interested in additional undergraduate research projects or master's students, PhD students. We have tons of data. We have thousands of samples. We may or may not be able to fund you depending on what happens over the next little while with our 2 biggest funders. But we're interested in any of that kind of stuff to hopefully move this all forwards. Thank you.
Okay, so I have a good news story. I know I'm sandwiched between 2 turtle talks, more or less, but these are toads. So I have a good news story about the Fowler's toads. This is a Fowler's toad, and I would point to it with a pointer, but I don't have one, so I will just merely indicate from a distance. So I'm going to present this, but really this, what's gone into this talk is the efforts of a whole lot of people. I've just put some grad students and postdocs in here. So I do have to recognize Victoria and Jessica and Natalie and Kat and Alex and all sorts of folks who have contributed to this. And if I forget to mention their names as being contributors to some particular item, sorry, but you know you did it, right? Okay. What now? No, I can't press that. I can only press what? There, I didn't even move.
It's all good.
I didn't move. Okay, so this is the toad. It's Fowler's toad, and we're here in Ottawa, as you can see. But Long Point is on the north shore of Lake Erie. It's one of 3 sites in Canada where the Fowler's toad still exists, and the place looks like this. This is a shot that Jessica took in 2017, a few years ago. And this is the kind of landscape that we got really used to throughout the first part of the 21st century. This is looking from the dunes at Long Point in the thoroughfare unit towards the lake. And on the is the marsh, except the marsh is all filled with Phragmites and the occasional person. As you can see, there are a couple of people in there. The toads hibernate in the dunes. They go and find a breeding site if they can in the springtime, generally in the marsh, if there is one. Afterwards, they migrate back over the dunes and spend the rest of the active season on the beach, hiding in the dunes at night and going along the beach. So it's a pretty simple affair. They migrate back and forth, and then they move along the beach all during the year. I've been studying these for really an enormously long time, going all the way back to when I did my doctoral work in the 70s. Okay, it shows how old I am. In 1986, based on a report that I wrote, Kasiewicz recognized Fowler's toads as being rare. That was the word they used back then. That got changed in 1990 to a new word, a couple of words, of special concern, without doing anything. And at the end of the 90s, based on an update report that I also wrote, was designated as threatened. And then Kasiewicz used the new criteria that they had to bring in, still threatened. And then finally, in 2010, also based on a report I wrote, I'm responsible for all of it, along with Alex Smith. It was designated endangered with a whole lot of criteria. Pick something from the B criterion, it's in there, as well as based on a population viability analysis, one of the very few Kasiewicz designations based on criterion E. So what happened to these poor toads? We've been studying them for a very long time, from the end of the 1980s until about 10 years ago. It was mostly work in this main study area, which goes from the end of what's the road Hastings Drive all the way to the other end of Long Point Provincial Park. And there are sites within there that we would survey. Since 2010, we've also been surveying in the thoroughfare unit, which is part of the Long Point National Wildlife Area, because we had some interventions put in there by CWS. So to catch a toad is really simple. What do? How do you catch a Fowler's toad? You go out at night, you go walk along the beach, you see one, and you pick it up. That's it. So they're great animals. It's fabulous. So we can measure them, we can photograph them, we can admire them, we can write down their GPS coordinates, we can get all sorts of data, and then we let them go. And the basic protocol is then to do it the next night again, and the next night after that, and on, night after night after night, until we keep getting the same animals all the time. And then we have a very, very good data set for calculating an estimate of population abundance. So I did I did it again. Come here, don't do that. Where is it? It worked. Okay. I think I need to go back. Come here. There. Come here. Okay, so in 1989, we did this protocol. We got, this is just males, because all amphibian biologists go into breeding sites and catch all the males because the girls hide. It's very smart of them. And so we caught, more than 40 males, and at the time they were designated as rare. This is about the time that people were worried about declining amphibian populations. So to my extreme embarrassment, my toads went up in abundance rather than down, and they were called special concern. Then they went down again. They went down, and then they went up again. They were called threatened, and by then we figured out where the girls were, so we could catch the females as well. They're on the beach, of course, hanging out, having a good time, because they're sensible. But then on, the population went down and down and down, and continued to go down. So what's going on with this trace of population abundance? Lots of data, really quite reliable. What's going on? In 2013, Dan Greenberg, and I based on Dan's MSc thesis, looked at the data up to 2011 to see what was going on. And we discerned that there's a breakpoint, a change in abundance from year to year. Before then, we could see evidence of density dependence, negative density dependence. The animals going up in order to go down, and it went down to go up. So it looked like that regime was going on. But beyond that, it's all decline, all the way down from there. So we looked at the usual suspects for this, some number of factors that may be responsible, including the toad zone abundance, the density dependence, the invasive Phragmites reeds that had come in, and by about early OOs had taken out most of the breeding sites in the marsh. We looked at the lake level, and we looked at weather variables, rain, snow, temperature, to see what was going to be significant. And we came up with this. We did the usual model selection thing with AIC, and we came up with a bunch of models. Over all years, the parameters that were turned up all the time were the log abundance of the year before, which contributed to the change, not the actual abundance, but to the change in abundance, are the rate of change, okay, the rate of increase, as well as the presence of Phragmites, more frag, fewer toads. So it looked like overall we had density dependence and very strong signal from the Phragmites reeds that had come in. Before the decline, it was all abundance, density dependent, no reeds, because the reeds had taken out breeding sites. And in the decline years, don't go back. No density dependence, but there seemed to be an influence of L of the lake that we didn't really think about that much at the time. We only had, not many cycles of change in the lake, so we didn't really think about it. Okay, sure, you ask, this is the ICC, what are the parameter estimates? How significant are these factors? And overall years, density and the frag, the weather variables, rain and snow, are minor contributors. Just add some more information, but not much significance. The pre-decline years, snowfall also makes a contribution. And afterwards, come here, don't go back. There. Lake level turns out to be extremely significant. Okay, remember the lake, all right? Remember the lake? Okay. Well, we zeroed in on the presence of the Phragmites because it seemed to be the most obvious and evident factor involved in the overall change in abundance in these animals. The Phragmites came in the mid-90s, and then it overwhelmed the breeding sites by about 2002, 2004. And a site like this in 1991, nice place for toads to sing in, turned into that in 2005. That's the same place from the same, take the picture from the same angle. A recovery strategy was written, as you have to do, and then a government response statement was issued, as the government has to do. And so the plan was to, come here, don't go back. Come here, go back. Fussy. Fussy. There. Okay. So to try to put in some mitigation measures. So Canadian Wildlife Service dug some ponds. They asked me, what ponds? I said, yeah, dig some ponds. So they built these pairs of ponds. These are one set of 6 pairs of ponds that we put in the thoroughfare unit. And these pens you see inside are from Catherine Yagi and her experiments in growing tadpoles at different densities. Worked great for the experiment, lousy for toads because they didn't like it. just when she started, grabbed toads and rescued them and grew them up. If you want to grow toad tadpoles, Jess knows how to grow them. And so along with doing her tests with the influence of tadpoles on ecosystems, grew up the tadpoles and then let them go, which makes sense. And so we did this head starting and rescue of eggs really laid in inappropriate places, kept them going. And finally, Ducks Unlimited and MECP and Environment Canada decided they were going to take, go to war against the Phragmites, and they were going to burn it, poison it, mash it, smash it with the marsh masters. And a place like this in the Crown Marsh, take a look at this dead tree, you'll see turns into that after they've mashed it. Okay, so did any of this work? Here's the trace of the abundance of the toads. Let's go a little bit forward and add ponds in those years, mash the frag, rescue the toads. Did this work? No, not really. But remember, I mentioned the lake. Here's the Lake Erie water levels. And as you can see, there are 2 rises in level, only 2, so not enough to really go on anything. But in recent years, it's risen again. And there was a floods in 2019 and lots of water around. So what do you think are the chances that the toads would respond? You think so?
Too much spectator.
Too much. Yes. Yes. It's good. It's good. So good. And it's really significant too. If you look at, if you incorporate the lag time between high water and high toads, it really is a significant relationship. How does it work? Well, these are drone shots taken by Victoria Tawa, 2018, before the high water. You can see this marsh full of frag. It's all overgrown. Just a few years later, the lake has washed in the dunes, has spread sand all through the marsh. You can see on your left, there's little squares of these pairs of ponds. And you can just see where they are on the other picture, still there. They are now obliterated. I mean, they're gone because the sand has continued to push in. And what you find now, at top is what the area looked like in the late 1980s, a few years after the washouts and of high water in the mid-80s. Great place for toads. This is what it looks like this year. It looks the same, and the toads love it. So this is what the environment looks like now in this year, and we went all the way along that edge, toads singing everywhere. In June, tadpoles everywhere, and little toadlets all over the place. So we've saved the toads, even without doing anything ourselves. So if you sing along, Rolling Stones song, right? You can't always give them what they want. Now, you can't always give them what they want. You can't always give them what they want, but if you try sometime, you just might find you can give them what they need.
All right, so good afternoon, folks. Thank you for joining us here today. For the last couple of years, we've had the opportunity to study the turtle populations down in Point Pelee National Park. So in the year 2000, Baylor stated there is no vertebrate group facing greater survival problems today. And over 20 years later, that still rings true. Over 70% of turtles are listed on the IUCN Red List, and over 60% of those are at risk of extinction. There are 8 Ontario species, and they're all at risk federally. So Point Pelee National Park is the first national park established for its biological importance. It is the most ecologically diverse national park in Canada, as well as the second smallest. Historically, it had the greatest turtle diversity in Canada. However, serious conservation concerns were evident by the 1970s. So this map here, you can just see the original extent of the marsh system, this line here. Now, so these arrows, everything south of those arrows is Point Pelee National Park, and all that kind of past marshland is now pretty much agricultural land. Cool. So Rivard and Smith conducted 2 surveys within Point Pelee National Park. At the time, they were the largest herpetological surveys in the park. They used hoop traps to capture turtles, and they captured 7 turtle species and noted a drastic decline of spotted turtles. However, it did describe the Blandings turtles as common. So Brown 2003, this is the largest turtle study to ever take place in the park. Data collection took place in 2001 and 2002. They used hoop and basking traps to capture turtles. They marked 1,599 individuals, however, saw no spotted turtles, and the Blandings turtle catch-breeding effort was over 5 times less than what it was in 1972. A shift in size structure suggested juvenile recruitment was limited for both the Blandings turtle and the snapping turtle. And they also did predator surveys and nest surveys, and they started the nest protection program that continues on today. So it's been going on for 23 years for the most part. I think, I could be wrong, but I think the only year it didn't take place was 2005. So between 2001 and 2021, or 2021, they protected 876 nests, and 505 of those were snapping turtle nests. So now into 2022 and 2023. So our research questions were, what is the status of Point Pelee National Park turtles? Has community structure changed? And has the nest protection program been successful? So we trapped in 2022 and 2023 using hoop traps and basking traps. And we set 2 sites concurrently and 4 sets of sites for a total of 8 sites. We had 3 or 4 visits to each site, and we had 13 or 14 trap days per site per visit. And we used a variety of different bait, so walleye heads, perch heads, carp, mullet, just kind of whatever, catfish, whatever we could get for free from loop fisheries. So the traps, we had 6 basking traps per site, and we had 12 to 16 hoop traps per site. Captures, so for each capture, we took standard measurements, and we marked turtles using notch codes, and we took carapace, plastron, and face photos. So we had a total of 6,527 trap days, and a total of 2,548 captures. So basically this table here just shows the captures, individuals, recaptures, and unknown. So unknown is basically, we know there was turtle there, and then unfortunately it escaped before we could process it, that kind of thing. So here's basically, so this is a painted turtle. On the y-axis, can you see the thing? Yeah, cool. So on the y-axis, we've got frequency, and then on the x-axis, we've got midline carapace length. The red is 2001 and 2002, and the green is 2022, 2023. And this kind of darker green, where'd the mouse go? I've lost, oh, there it is. This kind of darker green just shows the overlap. So this size structure does look somewhat improved compared to 20 years ago. You can see there's more younger turtles, so it would indicate that juvenile recruitment has increased. However, if you look at the catch breeding effort, it went down quite a lot from 72 to 2001, 2002, and it's gone down again into 2022, 2023. So that's not good. So the eastern musk turtle, there really hasn't been many captures for the eastern musk turtle, so there's not really a ton to say. Like there was only 2 individuals captured in hoop traps in 1972 and only 17 in 2001, 2002, and then we only caught 67 in 2022, 2023. However, it is interesting just that the side structure does look improved. So looks like there's more juveniles in our captures, and the catch breeding effort did triple. So that is really neat. So the northern map turtle, I don't really know what to make of the northern map turtle. The size structure looks really good, and it looks at least stable. The catch per unit effort for the hoop traps did double to what it was 20 years ago. However, we don't really expect to catch map turtles and hoop traps all that much. So I did put the catch per unit effort for the basking traps here as well. It has gone down a little bit, but kind of the general feel for the map turtles is that they're doing quite well in the park. So on to the Blandings turtle. I feel like the Blandings turtle here, this size structure histogram is somewhat misleading. It looks like the size structure has improved for the Blandings turtles in the park. But if you look at the catch per unit effort, it dropped a lot from 1972 into 2001 and 2002. And then it dropped another 20% over the last 20 years. And if you look at that value, that catch per unit effort value, that's actually the same value that we had for the northern map turtles, which is strange because we don't really expect to catch northern map turtles in the hoop traps with us baiting the fish, because obviously they're kind of more attracted to like mussels and snails and little bugs and that kind of stuff. But we do kind of expect to catch the Blandings turtles. So yeah, it's not good. Just kind of overall, the Blandings turtle doesn't seem to be doing very well in the park. But the snapping turtle, so as kind of mentioned earlier, the nest protection program is kind of biased towards the snapping turtle. So it is interesting here that if you look at the catch per unit effort, it did go down a lot from 1972 to 2001, 2002, but it has started to come back up. And on top of that, the size structure looks a lot better. So, which indicates there's more juvenile recruitment taking place. And that kind of shows us that the nest protection program is having like a desired result. We also did predator surveys. So for the most part, we did these weekly. We did them on Wednesday nights. We drove down the north boundary road of the park, and we drove down the west side of the park. So as you can see, in 2001, 2002, the predator numbers were quite high, as were the raccoon numbers. And in 2022, they actually went up for both raccoons and total predators. However, there was an outbreak of distemper in September 2022. And you can see like the numbers for the raccoons and the raccoons per kilometer and the total predators per kilometer just absolutely plummeted down for 2023. Which for turtles, I mean, that's kind of a good thing. So we also did nest surveys. We searched daily beginning late May. We found nests of 3 species in 2022 and 4 species in 2023. We discreetly marked the painted and snapping turtle nests, and we protected the Blandings and map nests. And then we just checked the boxes daily beginning early August. So you can see in 2022 how much higher the raccoons and total predators per kilometer was. And you can see with that drop in raccoons and just total predators in general, the predation rates of the nests dropped as well. So the key points here, basically, so this is very preliminary. Like we're kind of still collecting data, so just keep that in mind. But you can see the Blandings turtle here, like the catch per unit effort dropped so much from 72 to 2002, and it dropped again up to 2023. And for it to have that same CPUE value as the northern map turtle, that's just not good. And you can see it's actually now dropped below the eastern musk turtle. And I mean, it doesn't take long. In 1919, Patch described the spotted turtle in the park as equally represented to the painted turtle. And the last time a spotted turtle was seen in the park was 1994. So in less than 90 years, the spotted turtles went from the same as painted turtles to entirely gone. So It's pretty not good for the Blandings turtle. However, there is like good news here as well. You can see the nest protection is of course working for the snapping turtles. You can see that catch, breeding effort value come back up there. So for the Blandings turtle, I think basically step one is just kind of start figuring out where they're nesting. They often nest in the same spots around the same time every year or two. So yeah, like their clutch frequency is only about 0.48, but clutch frequency and clutch size increases with age. So yeah, if we just kind of put in the time and figure out where they're nesting and we can protect those nests, then hopefully we can help the Blandings turtle populations in the park start to bounce back much the same as the snapping turtles did. And acknowledgements. So obviously this kind of thing you can't do without a ton of folks. So just a big thank you to Rachel and Tara at the park for just kind of always being available. And Sydney and Teresa, Melanie, Kevin, Kaylee, Christina, Andrew, Devin, Maddie, and all the folks at Point Pelee National Park, and my field assistants, Taylor, Salem, and Dan. Can't do this kind of thing, obviously, without them. And Dr. McLaren at Lakehead and Lakehead University, and Todd Loop, and the Loop Fisheries folks for supplying the bait. Yeah. And there's pictures of folks and turtles.
Thanks to Greg's keynote from yesterday, we all know a lot more about map turtles. Medium-sized freshwater turtle, typically found in larger water bodies such as big rivers and lakes. A very aquatic species, so we would hope that they don't face too many threats, but of course, life's never that simple. And we know they do face threats such as motorboats and the spinning blades of death, otherwise known as boat propellers. And you can imagine metal boat propeller encountering the shell of a turtle causes some pretty severe trauma. Thanks to Greg again. Here's some photos from one of his other papers, looking at injury rates in map turtles at 2 of the study sites in Ontario. Some of these injuries are quite horrific. The black and white photographs doesn't show the horribleness of it, but to get some sense. So we know that there's about 6 million motorboats in Canada, which is a lot of motorboats. So the threat here is potentially quite large. There's been a couple of studies on injuries in map turtles from boats. Greg at his 2 sites in Ontario, Amanda, the Trent Seburn waterway, and injury rates at those sites were roughly between 4% and 20% of the population. But the real question is, are we dealing with a threat where map turtles, where there are very high numbers of map turtles and very high numbers of motorboats, is where we're seeing issues? Or is this a pervasive threat across the range of the species and those that we're at? Sean's talk this morning, remember, assumptions are gaps. If we assume the threat's everywhere, we're making a big assumption. We have to figure out if that's true or not. So we used the question, can we use iNaturalist data to assess the range-wide threat from boat propellers? I'm sure everyone's familiar with iNaturalist, community science projects, citizen science. People can report observations of native or exotic species. To date, there's more than 12 million observations on iNat Canada, over 37,000 species from almost 200,000 people. A lot of data. There's over 65,000 turtle observations, and the northern map turtle is the 4th most common species reported on the platform. If we look at the map of the map turtle according to iNaturalist, which is the top map, the bottom map is the Pacific map, just for Ontario. My apologies. When we compare the distributions, they're quite similar, so it suggests that the iNat data does represent the complete distribution. And the Quebec distribution, no offense, is not very big, just a little bit around Montreal, so we can just skip that. So our approach was to go through approximately 2,000 observations of map turtles. They ranged from 1979 to 2021. And from that, we identified approximately 1,500 turtles where we could assess the carapace in the photograph. And so we used photos where we could see all of the carapace, nice top-down shot, and also profile shots like this, where you're just seeing one side of the turtle. Now, that's if there's an issue, this because we can't see the other side of the hill, so to speak. We don't know if there's an injury on the far side of the turtle, but we included that data because most of the observations are like this, a basking turtle that can only see one side of the shell. So we acknowledge this is a limitation in the data. Next, we had to decide what is a prop injury versus injuries from other things. And we used the same techniques from other researchers, long cuts in the carapace, prop strike, V-shaped cuts in the marginals. These kinds of injuries are consistent with prop strikes. Sorry. These are considered prop strikes. Other things would be excluded. So what did we find? So this map shows all the points of map turtles. The blue circles are uninjured map turtles. The yellow triangles are injured map turtles. So overall, 4% of map turtles showed injuries that were consistent with propeller strikes. We then divided up the province into 6 regions and looked at the injury rate across the regions. Up here in the Ottawa area, injury rate was about 2%. Southwestern Ontario, injury rate was closer to 7%. So variation across regions. And we do have to be careful making comparisons among regions. There's a lot of variables going into this data, but nonetheless, we're seeing injuries in all the regions. So we pulled the data into a GIS program and then did a nearest neighbor analysis. We take every uninjured map turtle and calculate the nearest injured turtle to each observation. And from that, we get a sense of how far do you have to go to find an injured turtle. The range of the species in Canada is about 800 kilometers. So if it's a small number, it's going to turn out to be something in a few kilometers. And our median distance was only 5.3 kilometers, which is quite small compared to the 800 kilometer range of the species. We also wanted to look at injury rates within protected areas. So there were 16 protected areas, parks or national wildlife areas that had map turtle observations. And half of those protected areas had injured map turtles in them. The highest injury rate was in Rondo Provincial Park and the combined Long Point Provincial Park National Wildlife Area, which was up at 14%. And that's the same region that had the highest injury rate overall. We also wanted to look at where the injuries were on the shells of the turtles. And we found that there was twice as many injuries to the rear half of the carapace compared to the front half. And this difference was significant. So what can we take away from this data? So first of all, we'd like to argue that the 4% overall injury rate is likely probably quite a large underestimate. Remember back that most of the observations are turtles in profile, so 85% of the observations we're only seeing, 85% of the uninjured turtles we're only seeing one side of the shell, so likely we're getting an underestimate of that count. We're also obviously double counting turtles because we're not marking them. These are observations in iNaturalist. So if you can imagine some very public locations where there's lots of turtles basking, lots of observers, the same turtle can be recorded many, many times. Could someone close? Yes, thank you. If you can close that door, that would be great. And most importantly, we can't count the turtles that are killed by boat propellers. So if the turtle is hit and killed, it sinks to the bottom, it's not observed, it's not reported in iNaturalist. So we're probably really lowballing the number of injuries. One of the interesting results, we think, is the number of injuries on the rear half of the carapace. Other researchers have found the same kind of trend. There's a couple explanations as to why we might be seeing this. First of all, it's possible. Northern map turtles at the surface, motorboats fast approaching, and the turtle decides to do a dive straight down into the water column, something like this. Propeller is more apt to hit the rear part of the shell, and that's where we'd see the injuries. Other possibility is that injuries towards the front half of the shell are more apt to cause head trauma or spinal cord injuries, and turtles are more likely to die from those kinds of injuries. So the lack of injuries on the front half of the carapace could be because of the number of turtles being killed by boat strikes. Big question is, how do we reduce propeller injuries? Various people have suggested the obvious things. We could have more motorboat-free areas. That's probably possible in some locations, like provincial parks, things like that. There's good research to show reducing boat speeds and enforcing the reduction of boat speeds works in reducing injuries to large aquatic animals. And another possibility is the installation of propeller guards. And in fact, that's the real question that's kind of been bugging my brain for quite some time now. Why aren't boat propellers required on these outboard engines? This is not a threat that only affects things like turtles that we care about. It also causes injuries to people. These are just a few news stories I pulled off the web, including the one on the top about a 6-year-old killed by being hit by a boat propeller. I thought we cared about kids. Maybe we don't. I don't know. So you can go online, buy a prop guard for your motorboat, and you know what? You're not going to hit rocks anymore and break your propeller. There's a lot of benefits to the boat owner as well. And yet, no regulations for this. We've got regulations for seatbelts, but it's okay to cut people up with boat propellers. I don't understand this. So wrapping things up, 3 kind of takeaway messages. I think we've shown that propeller strikes are not just localized, but they are widespread across the Canadian range for this species. And that's valuable information for future COSEWIC reports that Nick's working on right now. We're finding injured map turtles within protected areas, and that is troubling. And the bigger message, I think, is that iNaturalist data is really powerful for answering some kinds of questions. And so it's very good in this case for looking at things like basking turtles, because there's lots of them, you can see them. Doesn't work so well for more cryptic species, obviously, but there's lots of possibilities out there. And I kind of encourage everyone to think about how they can make use of these free and massive data sets out there in their research for conservation. If you'd like to find out more about our research, we just had our paper published in the latest issue of HerpCon Bio. So it's online, open access. You can go home and download it tonight and read it. And I'll answer questions about it on the field trip tomorrow. If you've got any other questions, though, happy to take them. Oh, got to acknowledge the people that gave us money. Rogers Foundation, Clean Tech, Government of Canada programs of the Conservation Corps, and a big thank you to Greg Bulte and Amanda Bennett for answering all my questions about map turtles and boat injuries and propellers. And of course, a thank you to the hundreds of people who submitted observations of map turtles to iNaturalist. Thank you.